Hi, Riley. Can you hear me?
Yeah, sorry. I thought you couldn't see me for a second, but figured out it's working. So like the only technical thing they told me to look out for.
Hi, Kate.
What's up?
Oh, I was saying hi to Kate.
Oh.
Can I try to share my screen real quick just to make sure it works?
Yeah, for sure.
Okay. How good were people about sending their presentations to Jenna, just out of curiosity?
Pretty good, actually.
That's good.
Yeah, almost all. Yeah, it looks great. Well, actually, I think I only got 2 for this session, but the lightning talks, I got almost all of them.
Oh, great. You're busy.
Yeah.
Moderating.
Yeah. I also realized that there's a 30-minute window for questions between the first and second part of the lightning talks.
Oh, I didn't notice. I think just because there's not a Q&A between speakers, right?
Yeah. So hopefully there's lots of questions.
Hopefully.
Riley, this is Kate. Kate, this is Riley. Riley is currently working at the Sea Life Center as one of our interns and will soon be one of our research techs, actually.
Oh, good. Yeah, Riley stopped by our practice session this weekend, but I didn't know your affiliation, so good to meet you.
Nice to meet you as well.
So you were recruited by Jenna?
Sorry?
You were recruited by Jenna?
Yeah, forcibly.
Great.
No, there is definitely at least partial volunteerism.
No, okay.
Well, glad you're here.
No, that was a joke. It was by my own power, but yes, thank you.
So
I'm glad to be here.
you do bird work though? Yeah.
Sorry?
You do bird work?
Yeah. So I work with Jenna on a lot of what I do is kind of inputting data and data analysis. But then we also do the monthly bird surveys out in Res Bay, which is really cool.
So cool. I would say I'm jealous, but I also realize that it is November and winter's coming. So best of luck to you is what I'll say instead.
We're getting like 13 inches of snow tonight.
Meanwhile, I laughed because last week we got a cold front on Thursday and it was still chilly on Friday. And I went to an art walk downtown after sunset and I was afraid of getting chilly. So I realized halfway through the night that I had worn a long sleeve thermal top and a wool sweater. And I checked the temperature and it was 65 degrees. And I was walking around in a wool sweater. I'm in Texas, so I'm used to the heat. But I am from the north. It made me realize how acclimated I am to.
You lose your hardiness real quick.
For sure. It's going to be rough next year when I'm in the middle of the continent. So.
Oh, where are you going to be?
DC.
Oh, cool.
Yeah.
That's exciting.
But I'm gonna stay on the meeting, but I'm gonna black out for a little bit just to go through my presentation real quick.
Yeah, sounds good.
Thanks, guys.
And Riley, just so you get to know some people too. Well, I guess you met Kate already, but we went to the same undergrad and we had the same advisor. She's the one who taught me how to band gulls, which was awesome.
Wow.
Yeah.
That's crazy. Small world, I guess.
Yeah.
The lightning section is just a few minutes after this one, right?
I think it's like a half an hour actually. Yeah, we have a half an hour break. Busy day. So I guess we don't have to admit people, right? Or do you have to admit people from the waiting room?
I think you do.
Okay. So that's what you'll be doing,
Yeah.
right? Cool.
But am I like currently a co-host or anything like that?
Yeah, if you go
Okay.
down to the bottom and you click on participants, the window will pop up on the right and you should be able to see that it says co-host next to you. We're all co-hosts right now, I think. Speakers and moderators
Yeah.
can all.
So yeah, I guess that's what I'll be doing then, is just letting people in and making sure
Mhm.
stuff goes well.
Can you see my screen now, Riley?
Mm-hmm.
Okay, so it's the Waterbird.
Yeah.
Welcome slide. Okay.
Yeah, it's not in full screen or presentation mode, but I can see everything.
Yeah, I just realized that if I put it in presentation mode, then I can't see any of the Zoom things. So Oh, wait, there we go.
Yeah, that looks good.
Okay, cool.
Should I start admitting people from the waiting room?
I don't know, because they're not one of the speakers. Maybe we'll wait a little bit.
Okay. Yeah. It's called the waiting room, so.
Hoping our other speakers are going to be here soon. Although I'm realizing, I think that the other, there was another thing before this that goes right up until 1:40. So they might join right at the beginning. But Kate's here and she goes first.
I guess that's what matters then.
Mhm. Guess you can just start admitting people.
Okay.
Hello, everyone. We're just going to get started in a couple minutes. I know that there's a session just ending, so still have a little bit more time. For those of you just joining, we'll get started in just a minute. All right. Well, for the sake of time, I want to get started. I know some people are still going to be joining us from the other session, but welcome everyone to the Waterbird Society virtual conference. This is the behavior session, and we have a lot of great talks lined up. I'm going to stop sharing my screen. Up first, we have Kate Schlepper, and I'll let her take it away.
Thanks, Jenna. Sorry, I'm just getting my screen organized. Okay, thanks everyone for coming. Today I want to talk about the urban ecology of one species of wading bird, the wood stork. But I wanted to start by highlighting something that Juliet said in her talk this morning, which is that a lot of people in this audience are studying species that are efficient at locomotion over very long distances and are using habitats that are becoming urbanized. So in the next 25 years, it's projected that the amount of landscape in the United States that is urban is going to double from 5 to 10%. And this reflects a global phenomenon. So the United Nations thinks that by 2050, about 68% of the human population is going to be in a city. And so urbanization is a process we all need to be paying attention to, even if we have species that tend to like wildland habitats. And this is because urbanization is thought to be one of the main drivers of the Earth's 6th mass extinction event for lots of reasons. So there's, of course, the direct loss of individuals. So if you go and pave a parking lot, you, for example, might be destroying nesting habitat. But then there are all these indirect effects too. So things like changed noise and light regimes affect mating systems. It changes the way that animals move through the landscape. And so you're shifting where predators are concentrated. And so there are all of these changes that get compounded, and it becomes a problem because of sort of the pace and the scale of this change. And this is all summarized in this term HIREC, or human-induced rapid environmental change, that has been a large focus in the literature over time. And so one of the foundational studies in avian urban ecology is from Blair in 1996. He classifies avian species responses into these three categories. So you have urban exploiters, and I'm giving the example of one of my favorite species, a laughing gull. And if we're strict to Blair's definition, these are species that are actually more abundant in urban habitats than in wild habitats. So they're really capitalizing on urban environment to make their living. On the opposite end of the spectrum, we have urban avoiders. These are the species that cannot tolerate urban environments, cannot tolerate human disturbance. And so I'm showing a roseate tern because they're endangered in the United States, because we know that they're particularly sensitive to things like disturbance. And then there are other species that fall somewhere in the middle. So I'm showing a least bittern, but think about any species that uses agricultural lands, for example. So that means these birds tolerate some level of human modification to the habitat. They probably can deal with some level of disturbance, but they're not going to thrive in high density urban places. And so this is sort of the traditional way that we think about the types of response to urbanization. But the good news is, and of course we all know this, species can adapt, and they do adapt in lots of different ways. So if there's a change to the environment, species can respond by changing the time of their migration or the time of breeding. They can change where they move to, or they can change something about their behavior, or maybe there's a modification of physiology. And so this is one idea that I want us to keep in mind for the duration of this talk, that yes, we can categorize species into these neat little boxes, but I think we should bear in mind that species can and do change over time, and that even populations within species may be differently tolerant to urban environments. And so the species that I focus on is the wood stork, which in the United States in particular, because it's listed as a federally threatened species currently. The populations have undergone major changes over time. And so the question that I want to focus on is this question about whether storks have become better adapted to modified landscapes over time. So they were listed as endangered in 1984, but their populations are increasing, and I'll show you those data momentarily. Before I do, I just want to highlight that this is the red outline here is the entire range of the stork in the United States, the entire breeding range of the stork in the United States. And I'll be talking about storks at this population level scale, but I'll also be talking about regions. And in particular, I want to show you where the Everglades is in South Florida, because this was the historical core of stork's range. So there are coastal species, and historically something like 80 to 90% of the whole US breeding population would have nested in the Everglades. And so the Everglades habitat we can think of as ideal or pristine habitat for this bird. And this is what we've done. So this is what urbanization in the Everglades looks like. So in the foreground of this photo, I'm showing you the city of Weston. It is a high density urban habitat. And you can see it's separated literally by a road and a canal from the natural habitat. So this is the wild Everglades in the background. And so storks have inhabited all of this space over time, and in recent years are having to deal with the process of urbanization and the expansion of this urban habitat. And so what has habitat modification done to this species? So I just want to start by saying these hash bars are incomplete records. So in the early 20th century, we have a lot of single colony reports, but not whole population records. But from the 1960s on, when we do have good records, you can see that the Everglades population, again, this was the historical core for the United States, has not increased. So we protected this species in 1984 by listing it as endangered. And even though we've done a lot of management of the population over time, the Everglades portion has never recovered back to its historical highs. But the good news is the species overall is expanding. So Everglades population is still low, but the overall US population has increased to the point that in 2014, we were starting to hit recovery threshold levels, and the species was downlisted to the status of threatened. But one thing that was not known at the time of downlisting was the why, was the mechanism, what caused this increase in the stork population. And this is particularly interesting for us because the main stressor to this population, which is urbanization or habitat modification, has only got worse over time. So here's a map of Florida in 2010. So all the red spaces are urban habitats. And you can see the trajectory moving forward, whether we're really good or really bad about managing urban development, is going to be more urban development. And so this question about whether storks are better adapted to urban landscapes now than they were 30 or 40 or 50 years ago is a really important one, because we need to know whether their population is going to remain stable with this increased pressure in the future. So the main structure I'm going to use to sort of outline the rest of this talk is this basic premise of looking at what's changed, so we can have intrinsic changes. I showed you this graph before of things that can change within storks themselves. And we'll also talk about extrinsic factors. So what about the habitat has changed in the last 30 or 40 years? So what's changed? And then we'll talk through whether that change is good or bad for the population in terms of population resilience moving into the future. So just a very quick take on methods. I'm going to show you, I'm going to be using two different data sets. So first is actual fieldwork colony monitoring that my co-author Betsy and I have done for about 7 years. We were entering colonies and measuring things like reproductive success and diet. But I'm also going to be drawing on a range-wide population data set. So these are data from researchers all over the southeastern United States. I have data from 191 different colonies over a 17-year period, looking at both nest numbers and productivity. So I'll be showing you these maps as kind of a cheat as I show results, but just realize that I'm drawing on two different data sets here. Okay, so the first change that we have observed that I want to talk about is that northward expansion. So I showed you this graph before, all the bars were black, and here I've highlighted where the increase in storks, where we have seen the increase in storks. So the Everglades storks are in black, and again, we see no recovery over time. But the population has increased overall, primarily because storks have moved very far north. So as far north as North Carolina, which wasn't even historically part of the stork's breeding range. So the population overall is increasing because storks have expanded. And we have to follow up this observation by asking the question, is this change actually good? Are storks being productive in these places where they've chosen to expand over time? And it looks like yes. So first of all, we can look at nest numbers. That was my previous slide. That's a good proxy for sort of early breeding conditions. So what habitats are drawing storks to, are drawn to at the beginning of the season. But we need to follow that up with this question of productivity. Are they actually pulling off chicks? And when we look at the productivity numbers, we see good news. So here's the Everglades, the historical core. And we see that productivity in the north part of the range, where storks have expanded in only the last 30 or so years, is better. It is as good, if not better, as it is in the south part of the range. So far, this is looking really good. The change we've seen in storks is good for storks in terms of population resilience. The second observed change I want to touch on is this expansion into urban habitats. And when I say urban, I mean really urban. So here's a wood stork nest. And if you squint through the leaves here, you're seeing a golf course. And so storks are nesting in retention ponds and housing developments in county parks. So very high density urban habitats, they've sort of set up shop and are now trying to breed for the first time. And so since 1996, depending on how you want to define urban colony, we'll say 10 urban colonies have been established in just the last 20 or so years in the Everglades. So in this place that is supposed to be pristine habitat. And two key kind of notes here to help us think about it. Urban nesting takes place even when the Everglades marsh is in really good hydrological condition for foraging. And then in some years when the marsh is in really bad condition for foraging, urban colonies on the outside of the Everglades are the only places in South Florida where storks choose to nest. So this is telling me that this is a good thing, that storks are relying on urban habitats to sort of bed hedge in years when the marsh isn't doing well. And we can follow this up by looking at productivity. So here we have birds that breed in the marsh versus birds that breed in urban habitats. And again, we see productivity on average over a 7-year period is as good, if not better, than the marsh nesting birds. So it seems like urban habitats, the numbers of storks nesting in these places are very low. So storks still require the natural habitat, but they're doing well when they do choose to nest in these urban places. One quick caveat, I'm not going to spend too much time on. When we look at the regional data set, so this is more data over a larger spatial range, there is a little bit of question that arises because this purple area is the most urban part of the US breeding range, and productivity is on average lower than in the other places. But the caveat I want to give here is that these are productivity numbers for storks who choose to nest there. So on average, productivity is lower here, but storks are nesting much more consistently, whereas the Everglades, they're doing well, but there are many years when storks just don't nest at all. So this far southern population or this far northern population, it's sort of more of a boom and bust sort of phenomenon. So overall, urban environments seem to be good for storks. And then the final change I want to highlight, major change in the last 30 years, has been the establishment and an exponential increase in the number of non-native fishes that we're finding in South Florida. And storks and other wading birds are eating non-native fishes. So whether you look at this non-native species composition or whether you look at biomass, we are finding non-natives in the diet. In fact, some colonies are eating up to 60% of their diet is non-natives in some years. And there are lots of reasons sort of at the outset where we might say non-natives matter. So you can think about non-native fish altering the community dynamics of the prey base. So it's going to change where in the landscape prey may be concentrated. In this particular case, the non-native species I'm talking about are usually tropical origin. So if, for example, Central Florida gets a cold snap, you could wipe out a significant portion of your prey base in a short time. And then lots of studies in other systems with other bird species show that different types of prey, even different fish species, offer different nutritional value. So there are lots of reasons why non-natives could impact storks in either a good or a bad way. And I'm just going to go kind of at the highest level here. So we're going to look at productivity according to the amount of non-natives that are consumed at the nest level. And when we do that, first of all, again, we see here again reflected that the average urban colony productivity is higher than the average marsh colony productivity over a 6-year period. But then there's an effect of non-native fish consumption so that urban nesting birds sort of face this penalty if they eat too many non-native fish. I'm not going to go into the reasons here. And it's sort of an unknown at this point, but it's just to say that if we're thinking about population effects of urbanization, it seems like this non-native component of this process seems to be pretty neutral. So most birds aren't eating high levels of exotics, and it's really only affecting a small portion of the South Florida birds that are nesting in urban places. So when we put the pieces together, whether you're thinking about expansion into far northern parts of the range or urban areas, it looks like both in terms of nest abundance and productivity that this is a huge benefit to storks. Whereas something like diet, it doesn't seem to have a strong effect one way or the other, at least as far as we know so far. And so when we think about this on balance, we are confident moving forward that yes, storks are in a better place. They are more resilient to this urbanized process of urbanization than they were 40 years ago when storks were originally listed under the Endangered Species Act. But one really important note, and this is sort of the big take home I want to spend my last couple minutes on, is that yes, storks are still a coastal species. They require intact wetlands. So I say urban, for example, I say urban nesting is good. They're in very low numbers in urban places. But they are benefiting when they can access those urban habitats. And I want to posit that this is actually really important. So I'm thinking of storks as actually being sort of a unique response to urbanization, whereas Blair talks about birds that only do well in urban habitats, or birds that totally avoid urban habitats or can only handle something in between. Here we have sort of a unique response where storks both require the natural system, but also benefit when they choose to move into urban places. And so this is sort of a unique category. And I think it actually applies to lots of things other than storks. And I'm really excited to be presenting this talk to this particular audience because I think you all have your own study species in mind, trying to figure out whether or not it applies. And where, from my perspective as a wood stork biologist, I'm thinking this applies widely is this paper by Callaghan 2019 shows that of all avian species, water birds are among the most urban tolerant, partly because they're generalists. And so and this is a global analysis. So again, we have species that can move long distances in short periods of time. And I think there probably is a lot of bet hedging happening in these species. And so we can think of the urban environment in a little bit more dynamic of a way. So just to conclude, this is my final slide. A reminder that urbanization is a problem because of the pace and the intensity of the change to the landscape, but it's not true of all species in all places, and it changes over time. So where storks initially had, urbanization was initially detrimental to storks, they now seem to be recovering and adapting to that process of urbanization. And so wood storks are one good example of that. But I think this commuter response to urbanization may apply to a lot of other species of animals in general that have large home ranges and can move in and out of urban habitats sort of at will. So I just want to take a final 30 seconds to thank some of the Wood Stork working group. I think some of you are in the audience. They've been super generous about sharing their colony monitoring data with me, and I really appreciate that. And also a verbal thank you to the Waterbird Society. Part of this research was supported by the Cushionling Research Grant. So again, I thank you, and thank you to the audience.
Thank you so much, Kate. That was an excellent presentation. You have a couple questions in the chat, but I think we are right at the edge of our time. So if you can just answer those in the chat. And if anybody else has other questions for Kate, again, please go ahead and throw those in the chat. And those can get answered there. Up next, we have Hannah Mirando. Are you ready?
Yep, I'm all set.
All right, awesome. So we're going to have a presentation next on vocalizations in oyster catchers. And again, if you have any questions as the presentation is going on, go ahead and throw those in the chat. And if we have time at the end of the presentation, then we'll get to those then. Go ahead.
I can't seem to share my screen. It says it's disabled.
Jenna, I think I could because I'm a host. So if you go to share screen and hit the little arrow button, you can choose multiple participants can share simultaneously.
Okay, I think I might also have to make Hannah a co-host.
Okay, it's working now.
Okay, great.
Okay. Hi, everybody. My name is Hannah Mirando, and I'm an undergraduate student at Cornell University. Today, I'm really excited to be sharing my senior thesis project with you all, and it is about the vocalization variation within the oystercatcher family, or Haematopodidae. So animal vocalizations are a trait which we know can differ greatly among species in a single family. And divergence in vocalizations can lead to some really unique vocalization patterns across a species that's across many species that are related. However, species that are more closely related to each other don't necessarily have similar vocalization patterns. Sometimes species that are very distantly related can. So it's not always explained by relatedness. There have been studies that have focused on a couple of areas and tried to investigate why this might be. And one of the main areas is about the physical environment. So this has to do with the acoustic adaptation hypothesis, which basically means that organisms will adapt over time evolutionarily to have their vocalization patterns be most optimal given their habitat. Also, there's been work done exploring how morphology affects vocalization patterns across families and even just stochastic processes and how this can come into play as well. Overall, though, in the bird world, most of these studies have focused in on passerines or songbirds, and there's not as much literature that addresses these types of topics in water birds or coastal birds, and so that's part of why I had this motivation behind the project. So now a little bit more about why I chose the oystercatcher family in particular. A.J. Baker was a scientist who really laid the groundwork for a lot of what we know about this family in general. In the 70s and 80s, he piloted a series of descriptive studies that focused on several individual oystercatcher species. And these included the species in New Zealand, the African oystercatcher, and the Magellanic oystercatcher, which occurs in South America. A lot of his work focused on systematics, but he did do a bit about breeding biology as well. And these images to the right kind of exemplify some of the stuff he was doing. The top image is an illustration of some of the displays that Magellanic oystercatchers might do if they're disturbed near the nest. And the bottom image is some of the real preliminary stuff addressing the vocalizations of this species. And so we don't have a whole lot of literature on the vocalizations of oystercatchers in particular, but A.J. Baker was a big contributor to what we do know. And since the 70s and 80s, technologies have advanced a lot, both in the genetic world and in bioacoustics. There's new ways to record birds and new ways to analyze their songs as well. So my project is trying to build on A.J. Baker's work while utilizing these new technologies as well. So the oystercatcher family as a whole really is able to successfully occupy a wide geographic range. This map depicts the range of all of the oystercatcher species combined. And as you can see, although they stick to coastal areas, they still have really big ranges inland too, and they're found on pretty much every continent. But despite having a super wide geographic range, this family has managed to stay pretty phenotypically conservative. So their morphology and a lot of their physical features, such as size, shape, even like plumage colors, are pretty similar overall. They haven't diverged in crazy directions. However, since we don't have a ton of information about their vocalizations, we can't really be sure that they have stayed vocally evolutionarily conservative as well. We can't really know that. And so this is the core idea that my project is driving at. So my question is, are there structural differences in the vocalizations of the family Haematopodidae? And like, what are these differences if they exist at all? And if so, do they mirror phylogenetic relationships? So are oystercatchers in this family that are super closely related, are they showing also similar vocalization patterns, or are they not? And is there something else in play? So to start to get at my question, I turned to Macaulay Library and Xenocanto, which are two amazing databases where the public can submit their own recordings and I can access them for my research. So I took recordings from there and I dragged them into Raven Pro, which is a software used for analyzing birdsong. And so that's how I got my data. So out of the 12 oystercatcher species that exist, I narrowed it down to 5 species that I felt had the most robust sample size in terms of quality, quantity, and just appropriateness of answering my question in particular. And those species that I selected were black oystercatcher, American, blackish, Eurasian, and pied. For each species, I selected 3 recordings that I felt were of the best quality, and I took all of my data from there. Here is a phylogenetic tree that illustrates the relatedness of these 5 species to each other. So as you can see, the black and American oystercatchers are the closest, with the pied being the farthest from those. And so for this comparative study that I wanted to do, I couldn't just take the species' entire vocal repertoire and compare them against each other. It would just kind of draw attention away from those really like fine detail type of vocalization structures that I wanted to explore. So I had to find a homologous call type, which is basically a call type that has biological significance for all of the species in a similar way. And so I chose call type A, which is used by these oystercatchers as a way to alarm, or if there's a disturbance going on, they might make this call. If they're in a pair and they want to make sure that the other oystercatcher is safe, they'll do like this contact call. So it's all about alarming and disturbance. And this is a very typical example of what the type A call might look like in my recordings. Onto the variables that I chose. The first is high frequency, which is simply just the highest point of each call note. So for this first example, it would just be the very top of the call note. That would be the high frequency value. For maximum frequency, this means the frequency at the location in the call that has the most power. So here, it would be the place in that call note that's the darkest, because it means the oystercatcher was loudest at that point. Delta time is simply the total duration of the call from beginning to end. Number of inflection points is just the number of times that the call curves. So in this example, since the call notes only have one curve, it is just 1. But if you had a very wiggly type call that goes up and down, that would have a greater number of inflection points. So I took my data, I put it into R, I found the predicted means of each species for each variable. I also did a least sum of 3 squares test to find the significance of each variable when comparing the species to each other as a whole, and not just on an individual recording basis. And here's what we found. Maximum frequency does vary significantly among species. And what is the most interesting is that the American oystercatcher and the black oystercatcher, which are the first two that appear in the graph here, they vary significantly despite being the closest related to each other. So this phylogeny illustrates that idea. Here, the color of the nodes of each species illustrate the value of max frequency. And so if nodes have similar colors, they'll be similar to each other. So like black oystercatcher has this greenish tip and American has this dark purplish tip. So they're pretty much on opposite ends of the max frequency spectrum, even though they're the closest. And black oystercatcher, in fact, even has more similarities to Eurasian oystercatcher than American. So that's pretty interesting. Also, delta time was found to be significantly different among species. And here, looking at the phylogeny, we can see that black oystercatcher has this orange color, which is pretty similar to the pied oystercatcher. So even though black and pied are the most distantly related out of these five, they still happen to have the most similar delta time values on average. The variable high frequency was found to not be significant for these five species. And the number of inflection points was another variable that we didn't find significant. So for these two variables, we didn't overlay the means with the phylogenies because we felt it wasn't significant. So the main takeaways here are that maximum frequency and delta time values differ between species significantly. However, it doesn't, the patterns that we're seeing are not necessarily related to how closely related the species are, because with maximum frequency, the two closest related species had the most different out of the five of their maximum frequency values. And for delta time, the two species that were the most distantly related actually had the most similar values. So going forward with this project, I want to investigate why this might be. And I think that there's a variety of factors that might be contributing to it. First is habitat, because although oystercatchers typically are just hanging out by the coast, they also do move around a little bit for breeding or at different times of the day. And like you saw on the map, they do have some inland areas that they occupy. And so just as the acoustic adaptation hypothesis proposes, the different environments might require different vocalizations to be most effective. Feeding behaviors are kind of connected to that, and they do have some slightly different feeding behaviors among some species. I'm also really interested in this colonial versus non-colonial breeding comparison, and similarly whether they engage in any kind of flocking behavior at all. So for example, the Eurasian oystercatcher breeds in huge colonies, while in the northeast of the United States, American oystercatchers will breed in just a small territory. The pair will be in the territory and there'll be no other birds around. So this will have some significant implications on how the birds might need to communicate. If high density birds have to, call over the voices of other birds. Also, there's this really distinctive pied plumage versus unicolor plumage that occurs in this family. It's interesting because it's not like there's one branch of the species that has the pied plumage and the other branch has the unicolor. It's similar to what we found with the vocalizations. It's not related exactly to relatedness. It's pretty mixed across the board as to where this pied plumage versus unicolor plumage kind of crops up on the evolutionary tree. So I wanted to see if the plumage has any kind of correlation with what we found with the vocalizations as well. And lastly, I'd like to thank some of the organizations that have aided me with this project, especially the Cornell Lab of Ornithology and the College of Agriculture and Life Sciences at Cornell. They both provided funding for me through the Experiential Learning Grant. And also Zeno Canto, Macaulay Library, and Cornell University have all helped me with resources to further this idea. So thank you all for coming to my talk, and I would love to answer any questions you might have.
Thank you so much, Hannah. That was an excellent talk. I think I see at least one question in the chat. Erica Knoll said, nice talk, Hannah. How was the phylogeny created initially? And do you have plans to add the other species?
Yeah, sure. That's a great question. So one of my co-authors, Elliot Miller, helped me make the phylogeny. And so he drew from several sources of literature, namely, the name is escaping me at the time, but there's a really big paper that basically spread out the entire phylogeny of all species of birds. And so he was drawing on that really significant paper that came out a couple years ago. So he created that for me. In terms of whether I want to add more species, I would love to add more species, but I think that the sample size is kind of an issue for me in the recordings themselves, especially in terms of like quality and quantity. So if possible, I'll add more, but for now, I'm just sticking with those 5.
Okay, thank you.
Yeah, excellent. Thank you. If anybody else has another question, we still have a couple minutes. You could pop it in the chat, or you could also just unmute yourself briefly and go ahead and ask. Someone else said it would be interesting to compare the other vocalizations of the species, which you were saying, yeah. And Ian Nisbet mentioned European oystercatchers are becoming highly urbanized too, so relating to the previous talk.
Yeah, I've heard about that. I've seen pictures of them like nesting on top of buildings and stuff.
Yeah, I imagine that would definitely have some impact on the way their vocalizations work.
Definitely.
I think Tracy was actually saying the other vocalizations, like not just the alarm calls or the distress calls, whatever. So
yes, that's a great point. And I've looked into it a little bit. This is more of my preliminary data analysis, but I've identified several other types of calls. There's like flight calls are usually longer, and there's also across the board, a lot of these species do a very distinctive like territorial vocalization pattern, which if you've ever seen them courting, you might have heard. And so this call type that I chose seemed to be the easiest to get data from and the most relevant biologically for now. But yes, I would love to include other types of calls. I think that there could be a lot of interesting stuff there too.
Great, thank you so much, Hannah. I think we have to move on now. Again, that was an excellent presentation. Up next, we have Jenny Howard presenting on age effects on foraging. And I'll just let her take over from here.
All right, hi everybody. Thanks for logging on today. I'm really excited to talk to you about some of the research from my PhD. Okay, so we may be familiar with aging from a human perspective because we see aging in ourselves or family members. So wrinkling skin or graying hair as an example. We also see aging with the naked eye in our pets. But understanding the aging process in a long-lived seabird like the Nazca booby is a different story. We don't have any external cues to indicate the ages of these birds. I only know the age of these birds from our long-term study in the Galapagos. So this bird you see on the left is 6, it's a young bird. This bird in the middle is 14, it's a middle-aged bird. And this bird on the far right is 22 years old. It's a relatively old bird in our population. And it's only with these long-term studies of natural populations that we can study aging more rigorously. Aging affects an individual's overall performance. And performance can be composed of reproductive performance and also physically demanding or athletic performance. For non-human organisms, we're particularly interested in aging because it affects how many offspring individuals can accrue during their lifespan. and therefore it affects population dynamics. Reproductive senescence describes a pattern of late life decline in an organism's breeding success. Physical and cognitive ability associated with performance follows a similar pattern, with declines in performance occurring at later ages. Age-related athletic performance decline and underlying physiological decline could help explain observed reproductive senescence. Within a population of Nazca boobies that my lab studies in the Galapagos Islands, we see reproductive senescence or declines in breeding success at older ages. So in this figure I'm showing you on the bottom left, we see that age is on the x-axis and we have annual breeding success is on the y-axis. That's also the probability of raising a chick successfully. And at these really young ages, we see really poor reproductive success. That improves to middle age, and then it declines in later life. But we know little about how their athletic performance changes with age. My overarching question is, how does aging affect foraging performance in the Nazca booby? And my specific goal is to test for age-related changes in physically demanding foraging performance to connect to documented declines in reproductive success in Nazca boobies. So we study a population of Nazca boobies on Isla Española, the far southeastern island in the Galapagos archipelago. Here, the orange star on the map. This is a really long-term study. My advisor, Dr. David Anderson, has studied this population for 39 years. Males and females vocalize differently. Females honk and males whistle, allowing us to easily distinguish each sex. Each bird is banded as a chick with this metal band in the orange oval that has a unique number, and this allows us to follow the bird throughout its lifespan. We've banded the majority of boobies since they were chicks, giving us a large population of known age birds. To study how foraging performance changes with age in Nazca boobies, I targeted birds in 4 different age groups: young, ages 4 to 6, middle age, ages 11 to 14, old, ages 18 to 20, and oldest, which was the 21 to 25-year-old range. And these age groups correspond to changes in breeding success for both males and females. We tagged birds during the incubation period, when birds have eggs, to keep the foraging motivation constant. More than 700 birds were tagged with GPS loggers across 5 breeding seasons. We put the GPS loggers in plastic sleeves and use a special kind of marine tape to attach it to the bird's tail. We examined the effects of age on 7 different foraging traits calculated from those GPS loggers. The first is absence duration, the total time spent away from the nest on a foraging absence. Next is total distance, the sum distance between consecutive GPS locations. Time searching was the duration of time spent actively searching for and acquiring prey. We identified behaviors from the GPS data using a discrimination method called the expectation maximization binary clustering method. And I'm not going to go into those details, but that allows us to separate periods of time from searching from something like commuting or traveling. Next, we looked at 2 components of flight speed. The first is ground speed, which is essentially the distance traveled between 2 points divided by the time to cover that distance. Airspeed is the speed of the bird relative to the air. It's basically the ground speed, subtracting the component of wind speed and wind direction. Mass gain was the total mass a bird gained during a foraging absence. And mass gain rate was the mass gain divided by the absence duration. We analyzed data separately by sex because of sex-specific aging and breeding performance, but also because of sex differences in body size. Females are larger than males. By mass, measured here when birds depart for a foraging trip, females are 17% heavier than males. When we look at wing chord on the y-axis, females are larger than males, but only by about 3%. Wing loading takes into account the bird's mass and wing length. So a higher wing loading indicates that a bird has more mass per wing area. So females also have a higher wing loading, which is about 9% more than males. I performed analyses in 2 stages using linear mixed effects models. In stage 1, I evaluated how age influenced the 7 response variables, essentially trying to find out what is the shape of the age effect. Age here was a continuous variable, and we evaluated a model without age, a linear parameterization of age, a quadratic parameterization, and several threshold parameterizations. In this first stage, fixed predictors were wing loading to control body size, date to control within season effects, and breeding season to control environmental variation across those 5 breeding seasons. Random effects included in all models were the identity of the bird to control pseudoreplication, and a bin departure date to control any environmental variation during an individual breeding season. I compared the performance of models using AIC, and the top model in stage 1 was the model with the lowest AIC. In stage 2, we evaluated how environment and the interaction between environment and age explained variation in our response variables. I passed the age parameterization from the top model in stage 1 to stage 2. And I evaluated the effects of 3 fine scale environmental variables. So looking at that red box, I looked at sea surface temperature, chlorophyll A, which is a measure of ocean productivity, and cloud cover, because boobies might have a harder time spotting prey during cloudier conditions. And these fine scale environmental variables were averaged values across the Nazca booby foraging area from satellite products. Because of known correlations among our environmental predictors, each predictor appeared individually within a model. Additionally, we included interaction terms of age by each environment predictor, and the best model was selected as a model with the lowest AIC. I'm going to only show results from where an age effect emerged, focusing on the results for this slide and the next slide from stage 1. These lines you're seeing are from the model, the top models within the model set, with the red line showing the model predicted mean from the top model, and the gray ribbon showing the 95% CI from that top model. R squared that I report is the marginal R squared, which only incorporates the variation explained by the fixed effects in the model. So for this and the following slide, linear age is on our x-axis, and the respective response variable is on the y-axis. If you lose track of what sex I'm talking about, I've put the little sex symbol in the corner of each figure. So what I'm showing you here is early life improvement in male flight speed. We saw an increase from really young birds to ages 7 and 8 before a slight decline. So the males showed early life improvement for ground speed and airspeed. Only females showed evidence of late life decline in foraging traits. Females incurred longer foraging absences in terms of both time and distance after the age of 17. For example, a 24-year-old bird, which is the oldest bird sampled in this population, spent 17 more hours and traveled 250 kilometers further than a 17-year-old bird. And that's a really meaningful difference for these boobies. Because remember, when they're out foraging, their mate is home sitting on the eggs, and they're running low on body reserves. So with these longer absence durations, the greater risk a bird's mate will abandon. And we know that 40% of nest failures in this species happen during this incubation stage. I also analyzed how distinct foraging areas were by age and season. We found segregation between 2015 and 2016 in where they foraged. We only found spatial segregation in foraging areas by age group during the 2016 breeding season. Here I'm showing you young and oldest males side by side. So the orange points are 2015 breeding season, and blue is the 2016 breeding season. The outline is the 95% kernel density, and the solid polygon is the 50% kernel density. So the young and oldest males foraged in distinct areas in 2016. Notice that the younger birds, this figure on the left, they forage in areas farther south than the oldest birds. On this slide, I'm again showing young and oldest birds, but this time for females. The young and oldest females foraged in distinct areas in 2016. And notice that the older females forage in a larger area than the youngest birds. And that's consistent with the females incurring longer foraging absences in terms of duration and distance. When comparing the 2 years where we saw this large spatial segregation, it appeared that foraging was easier in 2015. That was during an El Nino with warmer sea surface temperatures. In 2015, birds had a higher rate of mass gain. And birds spent less time foraging in 2015 with those warmer sea surface temperatures. And this suggests that Nazca boobies perform better in warmer waters, at least while incubating eggs. And I say better here because the birds traveled farther, or shorter, which is better for the success of their nest. I'd expected that foraging performance would improve from young to middle age and decline in later life, matching the pattern of breeding success in Nazca boobies. But the foraging traits we examined didn't follow this simple pattern, matching breeding success. Overall, we saw stronger age effects that emerged for females and less so for males. We saw evidence for early life improvement in some foraging traits, primarily for males. And we saw late life decline for females, which seems tied to physical degeneration. And this isn't the only example we have of physical decline in Nazca boobies. Bite force in female Nazca boobies declines from middle age to late life. Taken together with evidence from breeding performance and bite force from older female Nazca boobies, these results paint a picture of enhanced physiological degeneration in female Nazca boobies. And these late life declines seem directly related to physiological degeneration in these older birds because birds are motivated to return to their nest as quickly as possible. So during the incubation period, if a mate abandons before the GPS tagged bird returns, Galapagos mockingbirds immediately predate these eggs. We've watched many mockingbirds roll eggs downhill and crack them open against a rock. During the chick rearing period, Galapagos hawks can capture young Nazca booby chicks that have been left unattended at the nest. Parents that leave their nest unguarded have lower breeding success, so birds are motivated to forage as efficiently as possible. Old females that incur these much longer foraging absences than prime-age birds would be motivated to return to the nest, but something about their underlying physiology limits their ability to keep these foraging trips short. The late life declines we observed in females seems related to female reproductive costs in this system. This population of Nazca boobies has a male-biased sex ratio with more males than females. So females rotate through mates, and some males get kicked out of the breeding pool for a year or 2 or 3. It's like they take a forced sabbatical. Females attempt breeding annually and therefore have higher reproductive costs than males. These higher reproductive costs suggest a life history trade-off where females put effort into reproduction at the expense of self-maintenance, leading to earlier physical degeneration in female babies. I want to thank all my funding for this research, so Wake Forest University, Department of Biology, Animal Behavior Society, and NSF could not have done this without their financial support. And then the Charles Darwin Foundation and the Galapagos National Park. Thanks to them for permitting us to work in Isla Española and logistical support. I also want to thank my lab mates from Anderson Lab at Wake Forest University and all the field assistants who helped with this data collection because this project could not have happened without them. So I'm happy to take any questions now, but also feel free to reach out later and ask me any questions you have. Thanks.
Excellent, Jenny. Thank you so much. Again, everyone, if you have any questions, go ahead and put them in the chat or raise your hand like Dave Moore has. So I'll go over to Dave. Go ahead, Dave.
Awesome talk, Jenny. Thanks. I was just from hockey, I know that I can buffer my declining physical you know, my athletic ability with experience and treachery. So I was just wondering, to what extent do you think the birds, like you're just looking at physical effects, but I'm wondering how much they can buffer through, like, you know, there's a greater emphasis on efficiency of foraging, or, you know, they might have to spend more time in self-care, like how much they can buffer that, and what age do you think that stops being a strategy or a option?
Yeah, that's a great question. Definitely, it seems like once these birds have gotten to middle age, they have a lot of experience with both breeding and foraging. And we see that in their breeding, where they have peak reproductive performance at middle age. But those old birds do have that same foraging and breeding experience that middle-aged birds do. So it seems like they are motivated to return to their nest quickly. You know, birds that don't return, their mate is more likely to abandon. So something is keeping them from keeping those foraging trips short. Like, I don't think these females are going out for like spa days on the ocean. They really are motivated to return to keep that male from abandoning the nest. So it's hard to say like what age you might see a buffering like you mentioned. But in this population, at least, it does seem like these older birds are experienced. They want to return back to the colony as quickly as possible. But they might not be able to capture prey as well, or maybe something about their eyesight limits what they can see under the water surface to be able to catch prey as efficiently.
And I think we have another question from Pat. Pat, you can go ahead and unmute yourself.
Yeah, I mean, Walt Reed back in the late 80s, early 90s looked at gulls, at Western gulls, and the older birds actually did just as well, both in foraging and in reproductive success. And the females would come back to the nest and stay there actually longer, and the absent times were shorter. So do you think that's just, all of this is just a function of the prey distribution, plus the birds' innate genetic makeup of how they react to some of that? Because in gulls, certainly the older ones can produce just as well, and their foraging is more close to home.
Yeah, that's a good question. It does seem to be something about this system. You know, aging results vary across seabird species and systems. So there are no aging effects for some species, but for Nazca boobies, where they have this really unique behavior with divorce, where females rotate through the mates, these females are incurring more reproductive costs. So they seem to be putting less into their self-maintenance and more into trying to breed, which is what is keeping them from maintaining that foraging physique, if you will.
So maybe it's more of a mate thing. Maybe it's more of the males maybe having more investment, do you think, into the pair bond, and then the females not rotating through, and that might make them not deteriorate as quickly?
Yeah, that's something we're definitely looking into in my lab group, is aging and that pairing of the males and females. And maybe it's that some nests we see like a middle-aged male that is paired with an older female, and they're able to buffer each other more. But we haven't finished those results yet.
Thank you so much, Jenny, for your presentation, and thank you everyone for your questions. Jenny, I think you might have a couple more questions in the chat, so you can go ahead and answer those in there. And we'll move on to our next speaker. So up next, we have Chip Westlow. Chip, are you here?
Yes, I'm here.
Okay, excellent. And you can go ahead and get started whenever you're ready.
Okay, Dave Moore is going to be advancing my slides. Anyway, I'm here today to talk to you about a seldom studied aspect of ring-billed gull behavior, And that's the spring reoccupation of the colony. And it covers the period pretty much from February to May of this present year. Next slide. This is what we usually think of when we see a ring-billed gull colony. You have hundreds, if not thousands, of gulls situated almost neck to neck, just barely outside of pecking distance. And in this particular colony, this particular picture is at Peninsula A at Toronto Harbor. There was about 4,000 ring-billed gulls here. So I'm looking at the process of the colony reoccupation. Next slide. I've been going to ring-billed gull colonies for over 50 years, and I've often thought about this question that I'm going to deal with today. And the colony here at Toronto Harbor is by far the most convenient. There are all kinds of limitations when you actually think about sitting down and going to a colony just about every day during the period of February to March. Access can be extremely difficult if you're dealing with an island. But anyway, in Toronto Harbor, you have an inner harbor and an outer harbor separated by a series of natural occurring islands. And then to the south of the outer harbor, you had a 5 kilometer long spit made from construction material that they've excavated in Toronto over about 40 or 50 years as well. We'll be focused mostly on what we call Peninsula A and Peninsula B. There are 2 other peninsulas you can see from the photo on the left. Peninsula C and D, they won't enter into my discussion here. Next slide. This is the current aerial view of that study area, Peninsulas A, Peninsula B, and then there are the 2 embayments, embayment A and embayment B, and those 2 areas, as well as the outer harbor, will be the main areas that I'll be referring to. Next slide. There aren't a lot of previous studies to examine and to tell us how other people have looked at it. Keys Vermeer had a monograph on ring-billed gulls back in the 1970s, and his observation, which was very short, simply said that when the gulls come back from migration, they have activity periods at the beginning of the day and at the end of the day, and they're not occupying the colony at all during the middle part of the day. So the 4 of us observers had bicycle access to the spit, and we set up 3 observation periods: early morning, midday, and late afternoon. An hour or 2 or even more around both the early and late afternoon periods. And we were interested in recording the number of gulls, the locations they were visiting, the behavior in those 5 areas. And the field time that we actually ended up spending was from January 16th to May 9th. We had 109 observation periods on 87 of the 114 possible days. Or in other words, we had somebody out during an observation period, at least one person for 3 out of 4 days for a period of 4 or 5 months. Total observation hours was 211, and you can see the distribution there of the observation periods, over 60% early morning and 20% plus or minus during both the midday and the late afternoon. Next slide, please. This next series of slides will be a time series based on this format here. I can tell you, or will show you, what happens as the period goes, as the time goes by. On the 1st of February, for instance, there were very few ring-billed gulls and lots of herring gulls on the north end of the outer harbor. The north end of the outer harbor was iced around the edges with varying amounts of water in the center. And the favorite loafing area for the gulls at this time was around that ice edge. And we noticed on the 16th of February that there was a huge influx of ring-billed gulls. And this sort of thing is noticed at many areas around Lake Ontario. The Presqu'île Provincial Park, for instance, they often notice influxes of 800 or 1,000 gulls on a given day. There's a big colony there. And you also see the same thing down at Hamilton Harbor. So by the 16th of February, at least a huge portion of the gulls had arrived. Next slide, please. On the 26th of February, we noticed that the ring-billed gulls had left this area at the very north end of the outer harbor. They moved away from the, you might say, the mixture of migrants and wintering birds, and they moved out from there. And we also noticed that these birds, the new ring-billed gulls that had arrived, were in perfect breeding condition, no head streaking, no remains of winter plumage, whereas the birds that had been there previously during early February, they were still in full winter plumage. So we certainly identified those as recently arrived migrants. And it was from the beginning on the 26th of February that we noticed that the ring-billed gulls, the breeding birds, were leaving the north end and slowly making their way down towards peninsulas A and B. In the morning arrival, they roosted overnight out on Lake Ontario, and they would come into the outer harbor every morning. Their areas of settlement would be on the water 200 or 300 yards southwest of the wintering birds, moving closer, like I said, down towards the embayment areas. Next slide. This is a shot then of embayment A in early March. The birds are congregated around the ice edge there. There were by this time, there were some already on the land, but I'll get to that in a minute. Next slide. This is what we quite frequently saw, and this is what we call a long line formation. It's a really thin, long line of ring-billed gulls. It's probably 1 or 2 or 3 gulls wide at most, but it would be 400 or 500 meters long. And there could easily be 800 or 1,000 birds in that long line. But they often sat offshore just like that, or came into the embayments and sat on the ice or the ice edges. Next slide, please. And it was on the 3rd of March that we first saw gulls come into land, actually land on the colony site where they would be breeding. And it was a hugely spectacular event, and that's what my video at the end will deal with. But so they were coming onto land, like I said, the first week of March. So it had taken them about 2 weeks, you might say, to acclimate to the area, to become familiar with it. You might also say work up the nerve to come back to the actual breeding area. We noticed a huge hesitancy in the way the birds moved to each of these next behavior situations, you might say, moving away from the winter flock, moving away from the open water and the embayments onto the land. And even to emphasize that, they landed there on the 3rd of March, but they did not land on the land a second time until 5 days later. So during the period from the 3rd to the 7th of March, they simply waited offshore all day and didn't come ashore. Next slide. This then is the rest of the season. On March 3rd, they already had arrived. So every morning from March 3rd onward, they would come, congregate in the water first, and then eventually jump onto the land. We had our first egg on April 16th. 2 days later, we had the scene that you see in the picture. There was a snowstorm on the 17th. The colony was completely covered with 3 or 4 inches of snow. And during the birds would still maintain that type of arrival, landing on the land in mass. They'd go to their nest, and after 1 day of returning to their nest, you can see that the snow has been cleared off the eggs, but the eggs themselves, the nests, are still surrounded in snow. And then we had the first instance of overnight roosting on the island, on the breeding area, on about the 6th of May. So there was a period here that we're looking at from the 3rd of March until, well, basically early May, where they were landing on the colony in mass every morning for a period of 60 days. Next slide. This is a summary then of those observations and of those movements. The birds started up in the north end of the outer harbor around the middle of February. They stayed there for a period of a week or 10 days, and then they gradually moved down towards the peninsulas A and B. Those big round circles represent where they would flock first thing in the morning. Like I mentioned, they came from out on Lake Ontario where they
Chip,
were.
I think we might have lost your audio.
Oh, really? Oh, okay.
If you can hear me still, maybe put something in there.
Okay, there we are.
Oh, excellent. You're back.
Where did we lose it?
Just maybe 30 seconds ago.
Oh, so you'd already, okay, I'd already been talking on this slide. Okay.
You know what? I'm seeing that might have been me. I'm sorry.
I won't hold it against you. Anyway, I'll start over a little bit. They come down from the north end. They come down from the north end of the outer harbor The large green circles represent areas where the flocks would congregate first thing in the morning when they come off the lake, and then they would swim down a little farther towards the embayments, and those arrows that are then going southeastward represent swimming on the part of the birds to get into the embayment areas. The red arrows indicate actual landing on the two peninsulas where they nested. Next slide. We were very interested in looking at the timing of landing on the colony relative to sunrise in the morning. And you can see the very first entry there under the 3rd of March. The birds landed on the colony 30 minutes after sunrise. But every day after that, they were there before sunrise. And you can see that, for example, in the middle of March, they were landing about 20 minutes before sunrise. In the first part of April, they were landing about 30 minutes before sunrise. And we get to the area time just before they landed there and stayed on the island overnight, they were landing probably about an hour before sunrise. And we looked at the same thing with respect to departure from the colony. The next slide, please, Dave. Here you have departure from the colony relative to minutes after sunset. And back on the 15th of May, when we first started making these observations, the birds were leaving the colony about 15 minutes after sunset. That duration continued for a couple of weeks. By the middle of April, you had birds leaving the colony about 30 minutes after sunset. And when you get down to the beginning of May, they were leaving over an hour after sunset. So what that is obviously, or what it tells us, is that as the season progressed, even though they haven't stayed on the island overnight yet, they're staying longer and longer and longer each day as you advance into the season. Next slide. So in terms of a summary, we have that it took the ring-billed gulls about 8 days to acclimate to what I call the local conditions before they started venturing away from these mixed flocks of breeders and wintering birds. We might refer to that as their arrival period, a period of about a week. And then there were 6 days that the flocks moved away from the mixed flock towards and onto the embayments, but not onto the colony yet. That was about a week or so during the end of February and the beginning of March. We might call that the approaching period, approaching the actual breeding grounds. And then there was 6 weeks of diurnal daytime residency on the colony, during which they were claiming their territories, finding a mate, copulating, building their nest, and laying eggs. This was a period of, looks like that's about a month and a half. We might call it the diurnal residency period. And finally, the last 3 weeks after egg laying, before they actually remained on the colony site overnight. And then, yes, this can be referred to easily as the 24-hour residency period. So in other words, it took 2 3/4 months, almost 3 full months, 11 weeks to develop this overnight residency at the breeding colony, going from roughly the middle of February to the first week or so of May. Now, I think my next slide is the video. Dave? It's a 2-minute video of one of the mornings when they came in to land on Peninsula B. This is more than an hour before sunrise. Oh, pardon me, it's probably 30 minutes before sunrise. Are you getting an audio with that?
Yep.
Okay. They're wheeling around in the sky like mad. They've come off the water, they're circling over the land. And this sometimes this goes on for a half an hour. And
I don't
now
know if the audio is coming through for everyone, Dave, but it might have just been the way that the screen was shared.
And the actual landing, there's a huge crescendo. They become very, very vocal. It's deafening. And then in a matter of seconds, they all land on the land, and you see them running to what might be their territories. That was really amazing. I mean, one of my assistants described it as a tsunami of the birds going across the land. Okay, and I'd like to express my thanks, our thanks to Andrea Creston. Backslide, please, Dave. Andrea Creston and the staff at Toronto Region Conservation Authority for encouraging the study and of course giving us permission. I owe a bit of thanks to Gail Fraser for letting us use her bird blind as a windbreak on Peninsula B. The winds in February and March can be very, very chilling, and any kind of protection was greatly appreciated. And I appreciate Dave for constructing the graphics in the presentation. Thank you.
Thank you so much, Chip. That was an excellent presentation and a beautiful video. We have a question from Pat. Pat, you can go ahead and unmute yourself.
Fine. So, Chip, is what you're saying is that these birds actually lay eggs and then leave them on the colony?
They do for sure. And in this case, it was at least 3 weeks. There have been a couple of studies that have looked at just that part of the reoccupation process. John Chardine's got a paper on it, and I forget who the other one is, but there are a couple of papers that look at exactly that thing, where they put thermistors on the, matter of fact, they've used artificial eggs with thermistors in them to record the amount of incubation they get during that period. And they definitely up and leave the colony and go roost on the water, and then come back an hour or so before daylight. They definitely do that.
Is the hatching success?
The hatching success, no, it's reduced. They compare it against maybe birds that are nesting a little bit later in the season after that period of departure has disappeared. And no, there's definitely a difference in the hatching success. It's not nearly as good.
And then one other thing, you know, that increase in the amount of time that they're on the colonies after sunset and before sunrise as the year progresses probably just is due to more of that twilight that happens as the year progresses, don't you think?
Well, it could be that, but it could also be, I mean, one of the advantages or one of the characteristics of colonial nesters is that they need that social stimulation from all their compatriots there. And I would imagine the frenzy just builds up like you can't believe. And I think that too increases the social aspect of their group behavior. So when they first landed, there probably weren't as many birds involved. And you saw that very first day, it was after sunrise that they landed. So I think there's a bit of a frenzy that builds up. And as a group, they are trying to get back to the colony quicker and quicker each day. But at the same time, they're very hesitant. There's all kinds of predators on the spit there, the Tommy Thompson Park. And we had coyotes and foxes and raccoons and skunks in the colony quite often. And I think they probably would have started their 24-hour residency earlier than they did. But there were many times when they were flushed by coyotes on the colony, and then they wouldn't come back until morning, or sometimes they would return as soon as the coyotes left. But there's certainly a coyote predation factor in there. And several people have looked at the impacts of, coyotes and predators on a colony. I'm not sure if they've looked at, this early in the year, but certainly during the course of the year.
Great, thank you, Chip. And that also kind of answers Ian Nisbet's question, which was, were there any terrestrial predators or other objective reasons to explain the bird's hesitancy to remain on land?
Well, there definitely is. I mean, I think they were watching for predators at the same time, and it would be nice to go to do the same sort of thing on a totally insular location. But I mean, getting to a colony offshore at that time of the year can be treacherous.
Yeah, absolutely. One more question. When do they establish territories? The way they ran after landing in that video seemed almost like it was pre-established.
We were watching for that, and there are certainly territorial battles almost as soon as they start arriving on the land. Now, I'm not sure if it's the same birds in the same area each time, or if they have new birds come in and try to take over the same territory. For example, when we were standing behind Gail's blind, right out 20 feet in front of us is where some of the birds were, and there were territorial battles going on there, I would say almost from the day one. But whether they were the same birds in each successive day, I did not know.
Excellent. All right, well, I'm not seeing any other questions, and it seems like we're out of time. I believe the next 30 minutes, we have a break in the schedule, and then the lightning talks will be up after that. So thank you again so much for all of our speakers. Those are excellent presentations. And thank you for everyone that asked questions. I'll remind everyone that there is an option to send people messages on the conference platform. So if you have any questions for the speakers that you think of later, always feel free to message them on the conference platform. And I'll also put the link to the silent auction again in the chat. So maybe during the break, you go over and check out the items and experiences that are available on the silent auction. There's a lot of great stuff there. Thank you so much, everyone, and I'll hopefully see you in some later sessions.
Thanks, Jenna.
Thanks, Jenna.
Thanks, Jen.